Hi, I'm Chris Brampton from Bio-Rad, and today I'll be presenting a poster on how to use the bio-plex apolipoprotein multiplexing bead array to test for apolipoproteins in our several diseased samples. Detecting changes in biomarker levels or even the presence of biomarkers in a precious sample can be difficult with traditional ELISA. Multiplexing magnetic bead assays afford not only a much higher sensitivity, but also sample conservation. In this study, we used an apolipoprotein multiplex panel to screen for some of the traditional apolipoproteins in the statin pathway. These include ApoA1, ApoA2, ApoB, ApoC1, ApoC3, ApoE, along with more traditional targets important in cardiovascular disease, neurobiology, cancer, inflammation, including ApoD, ApoH, clusterin, also known as ApoJ, and C-reactive protein. As you can see, we're looking at a lot of targets, and we're not going to use very much sample to do this. These serum samples are diluted 1 to 50,000, which means that we can use only 1 microliter per well to have a good idea of the levels of these analytes in our sample. We looked at 5 different diseases: traumatic brain injury, Alzheimer's, diabetes mellitus 1, and atherosclerosis, and sepsis, and then 1 sample for healthy controls. So we're looking at 10 different analytes, 6 different diseases, and 1 healthy sample. And this was all done on 1 96 well plate. And the samples were in duplicate. Clustering for observed concentration, we can see a breakdown of the different analytes in our samples and the different diseases. When we look at the actual levels more closely, we find that ApoD, ApoE, and ApoJ, the changes that we see are actually correspond to that published in the literature, with ApoD showing an increase in Alzheimer's disease, ApoE showing an increase in traumatic brain injury, and ApoJ showing changes in Alzheimer's disease, atherosclerosis, and sepsis, as has been reported. So we can confirm that the levels that we're seeing changing here have actually been published in the literature, showing that our panel is working as we expect. Serum apolipoproteins have been studied as risk factors in coronary health for a very long time, and one major finding has been the ratio of ApoB to ApoA1 can be used as a predictor in myocardial infarction risk. We have divided the panel between healthy and atherosclerosis samples, and in red here we can see ApoA1, the purple bar ApoB. And in our healthy set we can see the ratio between the two is quite far apart and much closer in our atherosclerosis sample, as you would expect these patients having an increased risk of myocardial infarction. Looking at this ratio across all our individuals from our disease states and healthy, we have found several of our subjects are actually at a higher risk of myocardial infarction, and interestingly we found in sepsis 2 out of 3 individuals have a higher than increased risk of myocardial infarction in the female, and in the male population 1 out of 2 has an increased risk of myocardial infarction. It's difficult for us to corroborate this because we have a limited amount of information from these patients because they were all anonymized samples. But it's certainly a very interesting point. And in fact, a number of the other samples that we have taken, including one from the Alzheimer's patient and another from an atherosclerosis patient over here, also show higher levels of increased risk or higher risk of myocardial infarction. So we can demonstrate that we have found all of our 10 targets in the plasma from individuals with several disease states, and that our magnetic bead based assay is very important, importantly can conserve precious sample. The serum was diluted 1 to 50,000, so only a very small amount of our sample was used for this multiplexing bead assay. We can use this as an important tool to discover biomarkers for different disease states, for things that you might not necessarily be looking at. I personally have used this assay in one of my previous experiments where we actually reduced ICAM1 in a prostate cancer cell line, and we found using a cell signaling panel that phospho-AKT was upregulated when ICAM1 was reduced. This isn't something we would have thought of looking at before, and I think it shows the value of this tool and the power of this tool when it comes to looking for things that you might not necessarily have thought about, and it could certainly warrant further research. Thank you.